So this next presentation is Dr. Doctoral research fellow specializing in inherited retinal diseases with a focus on female carriers of X-linked conditions like choroideremia. Her research addresses their underrepresentation in clinical trials and the complexity of their disease expression, offering critical insights into emerging therapies such as gene therapy. A strong advocate for equity in research, she leads innovative studies to improve diagnosis, care, and access for this often overlooked population. The CRF, and many thanks to all of you who have donated funds, have supported research of both of these, both Dr. Jolly and Dr. Gocuk. And as I sit on the research committee, it's always a hard choice because we're so acutely conscious of wanting a therapy now for our boys and our men that are affected. But this is a very important avenue of research in the scientific side to understand why that female carrier population is different within the population. And as we understand that little puzzle, it helps us to understand how choroideremia operates overall as a disease. So,
Hello, my name is Dr. Sena Gocuk, and I'm a research fellow at the Centre for Eye Research Australia and the University of Melbourne. First of all, before I begin my talk, I'd like to start by apologising for not being there to deliver this talk face-to-face, but I'm still very excited to be able to share the research that we do here in Melbourne, looking at female carriers of choroideremia. So my talk today is called Seeing the Carrier Experience, because I wanted to talk about how women are affected with these X-linked conditions and choroideremia, and also look into the reason why genetics alone doesn't tell us the whole story about the impact of disease for them. But before I begin my talk, I thought I'd talk a little bit about myself. So I'm a clinician researcher based in Melbourne in Australia. I completed my PhD last year, and you may have heard me give talks before, and my area of interest is female carriers of X-linked inherited retinal diseases, and choroideremia being one of them. And so my PhD very much focused on the clinical aspects of being a carrier, and how variable their presentations may be. And I was very fortunate enough to be supervised by 3 experts in this area, so Professor Lauren Ayton, Dr. Tom Edwards, and Dr. Jasleen Jolly as well. So I had great support behind me, which allowed me to achieve a lot of and complete a lot of projects during my PhD as well. So I know this audience knows quite a bit about choroideremia, and so maybe images like this might be something that you're familiar with. So I've got the right and left eyes of a person with choroideremia, and it's from an affected male as well. So what we see is these are fundus autofluorescence imaging, so areas that appear dark, so the black patches, are areas where the cells have died off over time, and the grey areas are the areas that still help with vision, and they are the functioning areas. So you may be familiar with how a male presents with choroideremia, but maybe less so about with women. And unfortunately, it's not as black and white as it is for males. So female carriers of choroideremia present with a spectrum of retinal disease severity. So you can have, from one end of the spectrum is the mild presentation. So when you look at the back of the eye, maybe you're not going to see any signs. It would be very hard for you to tell that the person is a carrier of choroideremia. Their vision might be very good. And if you didn't have genetic testing or a family history of the condition, you might even miss that they are, in fact, carriers. If we move up on the severity scale, the second is this mild to moderate disease, where you've got these patches of darkness that are surrounding. So they're not exactly the black patches that we saw earlier, but they're this grey mottled appearance. And so these can give us a hint as to the choroideremia or the carrier status of a female as well. If we move up, this is now considered moderate disease, and you can see just on the edges there, those black patches, quite small, that we saw in the earlier image I showed you. So those, again, are the areas where the cells have died off. But as you can see, quite a bit of the area is still grey, particularly, there's the central area over there, which we refer to as the fovea, or the macula, so the area that helps us with the central vision. That's still looking quite healthy, which is why vision may not be affected in this individual in particular, but the presence of those black spots, those regions, means that this person has moderate disease. And if we move up the severity scale to the end, that is severe retinal degeneration, and it's actually termed male pattern degeneration, because individuals with this kind of appearance is very similar to what we'd see in an affected male. So large regions of black on this imaging modality that we can see, those are the areas where the cells have died off, and just very limited areas of functional retina remaining. So this is the spectrum that was characterized before I started my PhD, and so we did a lot of work looking into different outcome measures or different tests that can give us more information about the impact of disease from a clinical perspective. But we were very interested in why some people can have mild presentation and some can have severe presentation. Even members of the same family may not present in the same way. So we delved deeper into the molecular mechanism to see the reason for this variability. And so that's what my talk today is going to be based on, to give you a little bit of an overview of the pilot study that we did in this area. So as I was saying, our clinical studies, found a lot of interesting things. We found that female carriers, when compared to healthy individuals or those with maybe milder disease, had reduced vision, they had reduced retinal sensitivity, they had thinning of the retina, so the back of the eye. And when we had a look at self-reported visual symptoms, so their experiences relating to maybe their central vision, their colour vision, side vision, their vision in the dark, or light sensitivity, female carriers reported that they had reduced or affected vision compared to those without retinal disease, so healthy controls. And like I said, to go to the reason for this variability that we see in carriers, we need to go back to basic biology and really look at the reasons for this. So if you remember from your high school biology days, you would remember that males have one X chromosome. And so if that chromosome is carrying the variant gene, they become an affected individual, or they have choroideremia. But women have two X chromosomes. And so if one of the X chromosomes has the variant gene and the other one has the normal gene, it can be quite variable, and that variation is due to a mechanism called X inactivation. So X inactivation is this process where during early embryonic development, each cell or cluster of cells decide whether it would like to express more one X chromosome over the other. So let's look at the situation where a cluster of cells, so let's say the retinal tissue, it decides that it would like to inactivate the variant gene. So that means it's expressing more of the normal gene. Then this can act as a protective mechanism and prevent against or protect against disease. So in this situation, a carrier would have relatively normal vision or be relatively unaffected on that disease spectrum that I was talking about. But if we consider the situation where the cluster of cells, so the retinal tissue, majority of the cells are actually inactivating or silencing the normal gene, they would be expressing more of the variant gene, and that would be leading more to retinal degeneration. So we hypothesize that this means that an individual with this scenario would have more moderate or severe disease. However, this idea of X inactivation skew doesn't always occur in a black and white manner. So it's not either all the cells in the retina are expressing the normal gene or all of the cells are expressing the variant gene. It's actually a relative, so it's a percentage expression between the two. And so if we have majority of the cells expressing the healthy or the normal gene, then we believe that these individuals are more likely to present with mild or moderate disease. Whereas on the other end, if we see the opposite, so most of the cells in the retina are expressing more the variant gene, then we expect those individuals, or we hypothesize that those individuals are the ones with moderate to severe retinal degeneration. So we were very excited in expanding on this work and looking at X inactivation patterns in carriers. But the first thing we needed to do was to find the tissue that correlates the best with the retina. So as you can imagine, we can't really grab a piece of the retina and put it under the microscope and have a look at which X chromosome it's preferentially expressing. So the next best thing was for us to find a tissue sample that we can access really easily. So something like blood, the saliva or cheek cells, and which of those is a good replacement for the retina. And so that's when we met a relatively healthy woman. So she's not a carrier, has no known history of an inherited retinal disease. And unfortunately, she had to lose her eye due to a cancer that was starting to affect her vision. And she was happy to donate her retina for our research. And so we also got her cheek cells, her saliva, and blood samples to answer the question of which peripheral sample best reflects retinal skewing patterns. And so for this work, we collaborated with researchers Professor Blewett and Dr. Gould from the Walter Eliza Hall Institute, who performed the lab-based assessments. So what we did, like I said, we had the retinal sample, we had the cheek cells, the blood, and the saliva. We extracted the DNA from all these different tissues, and in the lab, so Dr. Quinton Gould ran adaptive nanopore sequencing, where he was able to look at the whole chromosome-wide inactivation levels to see which regions of the chromosome are inactivated, and looking at it in terms of a percentage to give us the overall inactivation of each of those tissues. And so, like I said, we compared the skewing between different tissues. And even in the retina, what we did was we divided it up into quadrants there. So 12 o'clock, 3 o'clock, 6 o'clock, and 9 o'clock, and looked at the skewing at different regions. And it wasn't actually any different between the different regions, which you can see if we have a look at the peak of the purple graph. So that's representing the retinal samples. And then we had a look at the skewing ratios in the other tissues as well. And so we found the skewing ratio for blood, for saliva, and they weren't too close to skewing that we saw in the retina, but the closest was actually the cheek cells. So our study showed that if we were going to be replacing the retina with a different tissue that's going to give us the best idea of the skewing in the retina, it should be the cheek cells. The only limitation with this study is that it was only based on a single woman. So there is this is, a pilot study, and there is definitely a need for us to repeat this analysis with more women, which, I'll talk about in a sec, which, we are doing, but that is definitely a limitation that we will need to confirm these findings in a larger group of patients. But it was quite exciting for us to see that there is a sample that is easily accessible, non-invasive, and can give us an idea of what's happening in the retina. So in parallel, while we were doing the first component of the pilot study, we also performed a similar analysis in female carriers. So we didn't have access to any of their retina tissues, but we did have access to blood, saliva, and cheek cells. And so rather than correlating it with retinal skewing patterns, we were interested in correlating the skewing patterns of these tissues with what we see at the back of the eye, so from a retinal disease severity point of view. And so for this component, we had samples from 12 choroideremia carriers and 4 RPGR carriers. Most of the carriers that we included had severe retinal degeneration, and the reason for that is that it was a lot easier for us to be able to tell how much of a certain how many cells are expressing the variant gene compared to the normal gene. And we also included carriers with moderate disease and one carrier with near normal retina as well. And so indeed, when we had a look at our results, we found that majority of the carriers with male pattern degeneration were preferentially expressing their disease causing or their variant gene compared to their normal or healthy gene. The one carrier that we had with near normal retina was mostly expressing the normal or the healthy copy of the gene versus the variant gene. And we were also interested in seeing that carriers with moderate disease, so those with grade 2 and 3, were more in the neutral X inactivation pattern, which meant that they were close to having a 50-50 expression between the wild type and the variant gene. So they're also proving that those with moderate disease are not skewed towards one way or the other and therefore present with moderate disease. Now, I'd like to bring your attention to two sisters that we included in our study. So they're blood relatives, they're sisters with the same genetic variant. The difference is that the older sister presents with male pattern degeneration, so she's got severe retinal disease, whereas the younger sister has milder disease, and so she presents with relatively normal vision, very minimal visual symptoms, whereas the older sister is considered legally blind and doesn't have much of peripheral vision and has quite reduced vision. And so I've popped the X chromosomes back on the screen to help visualize what's happening with these sisters. So when we have a look at the X inactivation skewing ratios for the older sister, we see that 88% of the cells in the tissue are expressing the variant gene, whereas only the remaining 12% are expressing the normal gene. And so this regulation or overexpression of the variant gene is potentially what's causing the severe retinal degeneration in her eyes. Whereas when we have a look at the younger sister, we can see that 67% of her cells in the tissues are expressing the normal gene versus 33% are expressing the variant gene. So again, this upregulation of the normal gene is potentially acting protective in her eyes and so protecting her against severe disease, which is why she presents with this mottled appearance, which if you remember on that scale, falls into the grade 2 range. And so this case to me really emphasizes that even women from the same family carrying the same genetic change may have such different presentations, and the only thing that we can find different between the two sisters is their X inactivation skewing patterns. So the findings from our pilot study suggest that X inactivation plays a key role in influencing the disease severity that we see in female carriers, but we do need larger studies to confirm these findings. So first of all, we need more studies to find the best replacement for the retina. So in our study with the one healthy female, we saw that it was the cheek cells, but we need to repeat that test on a lot more women to definitively suggest a proxy for the retina, but also for the female carriers as well. So it'll be great if we had more relative groups that we can compare, like the two sisters, and also in general looking at the different disease severities and trying to, it would be great if we can say that this much expression of the variant gene will mean that they'll have at least moderate disease. So definitely larger studies are needed to confirm the findings that I presented today. And so that's what we are doing right now. So that's what my research is focused on right now. So my postdoc, during my PhD, I established the CREST study. So it stands for Carriers Research into Epigenetics, Symptoms and Treatment. And it's a registry of clinically and genetically tested female carriers of X-linked IRDs, including choroideremia. And so we're performing a range of clinical tests, as I described before. We're very interested in looking at how these conditions change over time, which carriers are more at risk of vision loss. And I know Jasleen is presenting on that work during the conference, but we're also obtaining DNA samples for analysis from these women as well. So at this stage, because we don't know what the best replacement for the retina is, we're getting any kind of non-invasive sample that we can get our hands on, and that our participants are happy to donate. And so this is a work that originated from Melbourne, but is not possible without the collaboration that we have in place with Perth. So Dr. Fred Chen and Professor McLaren in Oxford as well. So we're all working together to obtain more samples and perform these X inactivation analyses. And so we hope that this work will help us provide personalised care for female carriers, to give them more information about what to expect from their vision, how their vision may change over time, and to give them more knowledge about future planning and making decisions for themselves and future generations as well. And so I hope one day X inactivation may become a routine clinical test that's ordered by clinicians to guide in clinical care, also provide knowledge for better family planning, and also guide in treatment suitability as well. So in the space of gene therapy for X-linked inherited retinal diseases, I believe that X inactivation might give us an idea of how carriers will respond to treatment and whether it will help them with their suitability or eligibility for taking part in such trials. And I'd just like to end by saying that I couldn't have done this work without the wonderful team and support that I've received. So as I mentioned, my supervisors, Tom, Lauren, and Jasleen. I'd also like to thank our collaborators. So I've got a picture there of Associate Professor Marnie Blewett from the Walter Eliza Hall Institute, Dr. Quintin Gould from the Olivia Newton-John Cancer Research Centre. Down the bottom, Professor Robert McLaren from Oxford University, Dr. Fred Chen from the Lions Eye Institute, and also the wonderful team of people that I work with here in Melbourne at the Centre for Eye Research Australia, and particularly to the Choroideremia Research Foundation, who have been always very supportive of the female carrier work that we do and have funded quite a bit of the research that we've done in this space as well. And finally, I'd like to thank all of you for listening to my presentation today. I've popped my email on this slide, and I'd be more than happy for anyone to contact me with any questions or concerns that you may have. Thank you once again. Enjoy the conference.